Three, two, one. Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things
My name is
small.
Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC, dedicating aim to the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I have also since then worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen this week.
And at the end of the month, we'll be selecting a paper to do a deep dive on. So make sure to message us at MicroTWJC if something has caught your fancy.
Yeah, you can follow along with the papers that we discuss on either week in our shared Zotero library in the doobly-doo below.
Yeah, and we want to hear from you. So if you have any comments, please use the comments or tweet us at MicroTWJC. And boy, do we have a show for you today. Today we're talking about what happens when SARS-CoV-2 and flu infections happen at the same time. Then we'll have some talks about a revolutionary new way to develop nanobodies. We'll also be looking at things that researchers got wrong about spider silk. But that isn't all. How about bacteria engineered to destroy other bacteria? And the secret ways that bacteria talk to squids. And finally, a paper that you might lava. That is that's my pun of the day.
Good, great dad fun.
Yeah. Let's get started with firstly,
our only SARS-CoV-2 paper. So I did look for some other SARS-CoV-2 news this week, but I figured maybe it'll come out next week. Like in the US, they're gonna potentially approve the vaccine for ages 5 and up. But I couldn't find like a good thing to sink in. So stay tuned next week for I think there'll be some data next week we can go over.
This has been a press release week, really, rather than
yeah.
papers week.
But this is a good one, and this is great for flu season because, I mean, flu is like a classic, I think, co-infective virus because it is seasonal. It's always coming back. And there's good literature, like in staph, like it's well known that co-infection of influenza and staph, like it's just not a good outcome. And here, it looks like they're sort of diving into like the effect of, I guess, the vaccination, like immunity to either influenza or SARS-CoV-2 on that co-infection.
Yeah, this is a really interesting one because they use a mouse model mostly for it, but what they do is they infect mice with either influenza and then they add SARS-CoV-2 afterwards, or vice versa. And this is kind of to model like because we're just about to enter into a flu season, so I don't know if you this is a season where everyone's starting to get their shots at the moment for flu. And so they did these like kind of experiments looking at like mouse weight loss and how that is affected by these double infections. And what they kind of find is if they infect SARS-CoV-2 2 days after and 2 days after an influenza like infection, it's kind of like they stack up on each other.
Yeah.
So so looking at this weight loss.
Oh no, I was just gonna say that probably right. Like this is what like sort of makes intuitive sense because they're both respiratory viruses. So like you can imagine some sort of immune response that's too much right in the respiratory tract. Like that's not gonna have good outcomes necessarily if you get both at the same time. But it's not necessarily the case, I guess, for everything. It is seen in flu quite a bit.
Yeah, flu does a lot of like, I mean, because I remember like during the 1918 outbreak, there was a lot of investigations into like streptococcus and other kind of co-infections of flu because those spiked up at the same time as well.
Yeah.
And like so one of the main outlooks is like weight loss in mice. So when mice get sick, they stop eating, and that's kind of an indicator of how sick they are. So you can see like the SARS-CoV-2 has a dip around like a certain few days. And at the same time, like if you infect it 2 days afterwards, that dip in SARS-CoV-2 coincides with like the sickest point of
Mm.
influenza. So
Hmm.
there is a definite wrapping up effect, and if they space out the infections more, that it becomes less lethal,
Hmm.
which I found quite interesting. Um,
Yeah, I mean, I guess like
and
the right, there's something about or the immune response to influenza is not going to be the same as the immune response to SARS-CoV-2, right? They have different receptors,
right,
they're targeting different cells, they have different ways of interacting with the immune system. So yeah, it's like that spacing out is like,
yeah.
I guess there's time to recover from like one hit before they get the other. I don't know. Like these are just kind of like some random ideas that are flowing through my head looking at these.
Yeah, I mean, another thing to consider is that this is a mouse study, so they use like humanized mice, which have the ACE2
Mhm.
receptor, but and they have to do that because mice aren't generally that like they don't get ill from SARS-CoV-2 in the same way that humans do, so they have to give that.
Yeah.
Whereas influenza infects mice
Hmm.
readily. It is so. So the idea is that potentially these mice are just more susceptible to influenza anyway. So that might be something to kind of take into account for studies like this.
Yeah.
Um, but yeah, the other main takeaway from
There.
this is they also vaccinated
Oh,
them.
sorry. I just want
Sorry.
to say, but influenza is also like human influenza is not so great, I think, at infecting mice. So in this study, they use like a
Right.
mouse adapted influenza. So like there is a
Yeah.
well-worn like yeah, like I think when people study the human influenza, I think they do that in ferrets because of like
Yeah.
yeah. So
Ferret's a lot
yeah. So
closer.
actually, I mean, this could be like a major sort of like caveat that we should like highlight always with mouse studies, especially when you have two different viruses and you've had to like modify that model to accept both of them. Yeah.
Yeah. Yeah. And the interesting thing is that they investigated like vaccination. So what they did, but I say vaccination, but more like I think the correct term would be immunization. Because what they did was they gave them. So they didn't actually give them a vaccine. What they did was they gave them just like previous infection with SARS-CoV-2 or previous infection with influenza, just
Mm.
at a sublethal dose. So the idea is they'd give them a small infection. So this is more mimic like if they've had the infection before rather than if they had the vaccination.
Right. Yeah.
Um,
And we know this, right? Like I
and
think we've talked before that the immune response that comes out from a vaccine is different than the one that you get from a natural infection.
right, yeah. So yeah, I think what they found is when when they were like uh, when they co-infected them with the influenza, they found that protected the co-infected mice from death.
Hmm.
Whereas if they just vaccinated against SARS-CoV-2, that wasn't quite enough.
Mm.
So I think this kind of implies that influenza vaccination can be actually really protective in situations with this double infection.
Right, right. And maybe like yeah, more important than the than the immune response to SARS-CoV-2 in this instance. Yeah.
Yeah.
And I guess like that's
So I think
people
that this is
just like again, that's like a weird yeah, I guess it, I'm trying to think of like how to break this apart a little bit for folks. Like we always say like the immune response is specific, right, for the specific pathogen, but then there's also overlaps, right, in certain ways. So like this observation is trying to, or it is sort of signaling to us that the immune response to the influenza infection has protective effects, like uh, even in the SARS-CoV-2 case. Um, or there's always co-infection that they study. They don't.
yeah, it's always co-infection. They do also look at single infection. I, so I don't think that they found that the flu was much more protective for the SARS-CoV-2 infection.
That's
Um, so they had
the
some
black
like
line
that.
is
Yeah, the black
flu
line. Yeah, the black line
with the circles.
and the green line are basically the same. I think.
Mhm.
Uh, so I think yeah, the black line is just SARS-CoV-2.
Okay.
That with the so it's So, I mean, what I'd say is that since this is like an animal model, so you can't necessarily take it, extrapolate it fully to humans. But I think the idea that taking that load off the immune system for fighting off influenza will help if they in this co-infection situation,
Hmm.
which is something that could happen because there's still lots of SARS-CoV-2 around, and influenza's poised to make a comeback as well.
Yeah. Oh, yeah,
Um,
because people aren't isolating anymore, so we're not seeing that dip. I think we
Yeah.
saw that in a past episode, right? That like after regulations get lifted, like all sorts of respiratory viruses go up.
Yeah, and I think that there's a lot of news like now warning about influenza bouncing back. So I think there's a lot more push to get people vaccinated with influenza as well.
Hmm.
Um, so I think because it's probably going to be a pretty bad flu season if, and so the issue is if SARS-CoV-2 is like a value added on that, which
Yeah,
is we don't want.
yeah. And I'll say too that that this is also like an interesting like just for folks conceptualizing like what is like flu and SARS-CoV-2 together. Like it's almost as if we always say like pre-existing conditions like make you more susceptible to infections, right? Like flu is in some ways this pre-existing, right? Like it's a condition that
Mm,
comes seasonally and washes over the population, making them more susceptible to all sorts of things, right? Because that co-infection is not good, right? In many circumstances, not
mm.
just SARS-CoV-2. And so, yeah, getting the flu shot, right? Like in some ways, this is just saying like making sure that you're immunized from that it makes you stronger against whatever else is going on at that time. And specifically in this case, they're highlighting SARS-CoV-2. Yeah.
Yeah. So now we're going to go away from SARS-CoV-2 a
Oh, I
little
was gonna
bit
say
to look at.
minimal
Sorry.
put a plug in just to say like don't forget that SARS-CoV-2 also might be in the gut. But but
Yeah.
but I do
We.
want to say to that like when we think of serious outcomes to SARS-CoV-2 disease, right? That's driven by like ARDS, right, which is in the lungs. So, like, there is good reason in my mind to classify SARS-CoV-2 right as a respiratory illness because that's the mechanism of injury that we see for mortality.
Yeah. We're still unclear about what's going on in the gut and whether that could bloom back into a full infection or not. So I'm still like watching because I know
Oh,
that there's a group of
yeah.
scientists who are still working on that.
Sorry, so it's not just
So
gut, but that
that's
makes sense. Yeah, I forgot. Like, yeah, you're right. Like, it's also this like right, and maybe that's like from the standpoint of pancreatic cells. I was actually thinking like we saw the neuronal stuff, right? Like it can
right.
infect the olfactory epithelium and get into the brain as well. But again, like the
Yeah.
effects of those things are like people haven't seen that type of like mortality morbidity from that as much as we've seen from the respiratory. But you're right, I guess like yeah, he's like listing off some
And
different things.
it'll be very hard to actually find out the effects of that while there's lots of respiratory transmission going on, I think.
Hmm.
Because think about that signal might be quite low compared to respiratory transmission.
Yeah.
And so if you see someone who has a case with where it comes from the gut or the neurons or something that was very dormant, then we wouldn't. It would be very hard to pick them out because they might have got exposed to someone who had SARS-CoV-2 just by coincidence.
Right.
So it's very hard to pick out those kind of very rare
Yeah.
instances.
But we've seen from the
So
animal models, right? And like and also biopsies of people that it does get into those areas, right? So the effects are there, but
yeah,
in terms of, I guess, I mean, I don't think we really covered any papers. They might be out there, but specifically looking at those other effects of SARS-CoV-2.
yeah, long COVID is a very difficult subject, and
Yeah.
it's so I've been trying to look into papers, but a lot of it is like on stuff that is on measurements like that are to do with like psychometrics
Yeah.
and that's a very tough science to get into if you're an outsider because there's a lot of like clever social science stuff they need to do in order to pick out what the effects are,
Right.
and it is very vulnerable
Yeah. But
because I know it's very
oh no,
sorry.
I just want to say like I think that this is a good discussion from this sense of there's one thing to see like all this mechanistic information right down at the cellular level
Mm,
of viruses getting into certain cell types, but then like you also have to ask like the clinical relevance of those things. And I think one
yeah.
of the things that we're trying to say here is that like the ARDS thing is like this easy clinical relevance thing that like got pushed up and like in some ways people have been blinded to just be like this is what we're stopping, right? And like we're not seeing these things, so everything's fine now. But like, yeah, the science hasn't addressed the idea of like, well, if it is in the gut, then and it maybe has other long-term effects. Like that is allowing for the circulating virus to remain in the population, and like then what are we going to see, right? And like co-infection, that could be an issue, right? Like if that's the case.
Yeah.
Yeah, it's just like
I mean, fecal oral transmission. I mean, when you've got airborne transmission as well. So let's say you go to a restaurant, someone has not washed their hands, but they're also like have COVID. So it could be that you have to rule out like respiratory, and that it's quite tough. I don't envy people trying to find, because at the moment I've only seen one lab that have actually seen like who are really focusing on this,
yeah.
and it'll be quite interesting because there
Yeah,
is a
no,
like ongoing question of like
for
yeah.
sure. And I just want to say, like, I do agree that pancreatic beta cells have huge clinical relevance. I'm just saying that, like, the the reports of, right, like, oh, metabolic dysfunction or whatever, right, and like having that take a bad clinical turn, as opposed to the ARDS, like seeing those relative numbers, right? And then also putting vaccination over that and saying vaccination is going to reduce these. So maybe we'll see more signal from people that are having like these other adverse events coming from pancreatic. Like I just haven't seen that yet. If you have those papers, like we'd be very interested. We'd be interested
Yeah,
to review. Yeah.
I mean, it's interesting because I mean, this is almost like something to like Streptococcus pyogenes, which also can be an airborne like infection. People think of it as respiratory, but also has skin. It also like goes through different organs. It causes like heart difficulties. It's very hard to classify it. So some people tend to classify it based on diseases it causes. I mean, there are lots of different like infections that aren't just limited by one
Right.
transmission route, and so you can get lots of different
Right.
variants. And so I can so yeah, I'm just looking at minimal's comments. Yeah, it is possible for like, because sometimes when like SARS-CoV-2, especially when it goes systemic, that means it can really like have a lot of effect on different organ
Mm-hmm. Mm-hmm.
systems. Um, so I think thinking from an epidemiological perspective, people tend to classify it as a respiratory
Mm-hmm.
virus because that's the important thing when you think about controlling
Yep.
transmission. But when you're in a clinical setting, you're not just looking at lungs, you're looking at all the
Mm-hmm.
organs and you're looking at all the kind of things it can affect. So it's really like kind of, uh,
Yeah, I mean
uh,
the
well,
management
I
will
mean,
be, and like I guess the thing is like unfortunately, like based on the way that like policy has gone, I guess it's like, to me it's also, it speaks a lot also to the Delta variant's increased transmission rate. Like I heard in the news, or maybe I should have picked that news article, but like that New Zealand is no longer pursuing like a zero COVID strategy. I don't know.
yeah, yeah,
Yeah, I don't know if folks remember, we like covered a paper that was saying that like taking a zero COVID strategy is like the best thing that people could do, but it got abandoned by many countries, and it puts us in that same realm of like kind of like malaria, right? Like right there was that mirror in history, right? Where it's like at the
mm,
beginning of like trying to control malaria, it was like, let's do the zero malaria strategy. It's going to ensure the most amount of lives saved. But then, like, there was resistance to DDT and mosquitoes. People didn't want to use them, right? Like some of the strategies people used failed, right? And then they were like, okay, I guess we just have to minimize harm. And I do think that we're entering that era, right, of SARS-CoV-2. It's like, we guess we can't actually control it 100% in the world. Like we're going to minimize harm as best we can. And again, that's the focus on respiratory, I think comes like is driven by that. But like as better clinical descriptions of other potential clinical pathologies emerge for SARS-CoV-2, like
mm.
that will hopefully expand people's awareness and like their willingness to implement strategies to reduce those risks as well. Um, but yeah, I guess we should see
Yeah,
what come. We'll definitely be looking on Twitter to anything people tweet to us. Yeah.
yeah. All right. So next paper is called ssDNA recombineering boosts in vivo evolution of nanobodies displayed on bacterial
Hmm.
surfaces.
Yeah, so this is like from the depths of synthetic biology for sure. So we've talked a little bit about single-stranded recombineering in the sense of like it's a different type of technology people can use to get mutations inserted into things. And one of the strengths of it was that you could make like little constructs. The constructs were more minimal that you could put inside of
Hmm.
the bacteria, And then I'm not sure if we talked directly. We talked a little bit some direct evolution. We talked about nanobodies, right? Those are smaller antibodies. They're made from camel. They're the
Mm.
the binding
Yeah.
region of camel antibodies. Um,
Yeah, they alpaca
yeah,
punch.
alpaca punch. I thought you were gonna need one one pun. You're gonna bring another one out right there.
I'm not a one pun man. You put a penny in me. I can't stop now.
um, so in this paper, they're bringing together those single-stranded recombineering strategies, right? It makes it easy to just put this like single construct into a bacterial cell, and that gives you this whole library of variation. But they're combining that with nanobodies displayed on the surface of those bacteria. So essentially, the library that you end up creating with this strategy, right, has a different nanobody on the outside of it. And I'm not sure if people remember talking about phage therapy. We definitely didn't do it. It's not phage therapy. Um, phage. Well,
Phage display.
phage display. Yeah,
Phage
phage
display. Yeah,
display.
that's I think we.
Yeah, that's one of the early directed evolution sort of setups where you're selecting for the whatever it is that binds to your pressure. So like you want better nanobodies that bind to I don't know, like Epstein-Barr virus antigens, right? So you have a column full of those antigens, and you flow through your mutated your library of mutated bacteria, and the ones that bind best are the ones that you send into the next round of evolution, so that you hopefully get better and better binders. So this is just the figure that you got pulled up here is I guess the construct, right? The evolving
Yeah.
evolution platform. You have a bacteria that has this particular single-stranded recombineering plasmid in it, and in the genome there's a membrane-bound nanobody, right? That's sitting there, and through mutagenesis and through purifying out, you'll get better binders displayed on the outside of these bacteria, and you can send that through another round of mutation, and yeah, just get better and better nanobodies on the surface.
Yeah, I think we've done like similar studies to this before, like that use CRISPR to incorporate stuff. This one
Yes.
used something called red beta, which is like a mechanism for like repairing like the genome. So essentially, if like a strand gets like snapped off, red beta comes along and kind of like puts it back on, so it anneals the DNA to another DNA.
Hmm.
And basically here it uses that to anneal like a different version of the nanobody gene. So they got the CDRs, complementary the bits that grip onto whatever it's binding to. So complementary determining regions.
Yep.
But I just think of them as the grippy bits generally. And yeah, it uses like they generate a load of variation of these like in and put them into a, like I think they stick them into a plasmid or they stick them into the E. coli, and
Hmm.
then they stick the variants in this plasmid that go in, and these little ssDNAs go in and interfere with the original gene, and then you get these E. coli that have all these different genes, and then you can just like sort them out by having like their target on like magnetic beads,
Hmm.
and so they got magnetic activated cell sorting they use to filter out all the E. coli that have the nanobodies that bind the most, and use that to filter out. They take out the genomes, figure out which ones are working, and then they roll it through this process over and over again until they get their best nanobodies.
Yeah.
Um,
Um,
so
yeah, no, that's
yeah,
that's what it does. I mean, later figures in this, like I think, I guess probably scrolling to like um, figure 5 or something like that,
figure five.
right at the
Let's
end.
go. Gotcha.
Yeah, just
The
shows
stru. Oh yeah,
it just shows us that like through these evolution experiments, right? Like they picked up these various mutations, and these mutations are better at binding. Um, yeah, better at binding their
yeah.
target.
So they got this one protein off the surface of enterohemorrhagic E. coli, which is
Hmm.
a pretty nasty E. coli. There's a different flavor of E. coli called EPEC, which is enteropathogenic E. coli, and they both have this kind of outer membrane protein that this nanobody could. So initially it targeted the one on EHEC,
Yeah.
and they wanted to evolve it to target the next one. So what you can see here is a comparison of different mutations targeting both. So obviously, like there are a few intermediate mutations, and there's like
Yeah.
so the original mutation is TD4, and it binds like perfectly to the original one, but doesn't bind the other one very well at all. So you've
Yeah.
got, and then with each mutation, you can see
They get
that
better
they
binding. Yeah.
yeah.
Yeah, and like
So
this is their toy, right? Like I think we've spoken about this before. Like all like technology sort of development papers will have their little toy example, right? But the idea is that this is a proof of concept that extends that to like you can imagine your favorite protein, right? That you want to evolve specific nanobodies towards, and then you can use this method to get those better binders.
yeah, like I mean, the key one everyone's thinking about is like spike at the moment, but then
Yeah. Yeah, I'm trying to
there are various
figure out
like
what was we
cancer
did something in the
proteins or like
I feel like we talked about nano. I think we talked about directed evolution of nanobodies against
I
spike.
think we have.
Yeah, nanobody directed on
Uh,
7.4. We did one, and it was continuous directed evolution. They were using yeast to do that. So
yes.
they had their mutation system sitting inside of yeast, and very similarly, I think this is what we said during that deep dive was that in some ways they're reconstituting the way that the human immune system works, right? By making sure all that variation is sitting inside of a library that they just select out of and
Yeah.
then try to iterate over. So this is the same. This is a very similar experimental design or like conceptually, right? Like design space, but we're doing it in E. coli, which maybe I don't know, easier to transform than yeast. I think yeast are really easy to transform these days. Um, yeah,
Yeah, so much technology is out there now. It's amazing.
yeah. All right, that's all I think I want to say about
Right.
that one.
Yeah, same. Next one is human commensal gut proteobacteria withstand type 6 secretion attacks through immunity protein independent
Hmm.
mechanisms. So, main thrust of this paper is cholera is bad. So cholera, it's bad bacteria. People don't like
Yeah,
it.
don't drink it.
0 out of 10.
Don't drink it.
Uh,
Yeah.
um, and the thing about cholera is one thing that it does that is, I mean, it does many things that are bad. It secretes like this cholera toxin that causes your gut to have diarrhea,
Yeah.
but
Lose a
also
lot of
like
water.
attacks your gut microbiome directly
Hmm.
using a type 6 secretion system, which is like a little like injection system where it injects toxins into bacteria that's near to.
Hmm.
So when the earlier stage of infection, it's a pretty like nasty thing for it was a way for it to establish its niche
Yeah.
in the
Yeah.
gut.
So yeah,
And so this
let's. I just want to expand that a little bit because we haven't spoken. Like I think we brought up a bunch of typeface papers, but it's nice to expand that. Like the goal of cholera is to grow in your gut and then get pooped out at really high volumes.
yeah.
But your gut already has tons of bacteria in it, and they would not like to be like pooped out in high volumes necessarily. They're happy living
Yeah.
in close proximity to the gut walls, right? Like they found their niche. And so type 6 secretion like could be used to clear out some of those things like these are protective good gut bacteria you can say right are being cleared out by cholera so that it can start growing in there and then that toxin can instigate like that watery stool and then it can spread everywhere so yeah that's I just want to say like that's the scenario cholera is trying to enter inside of the gut.
Yeah. But the thing is, like in lots of other animals that have cholera, so cholera infects water supplies, and lots of animals drink them, and a lot of them don't get the same symptoms that humans do.
Hmm.
And there is this theory that part of the reason that for that is because they've got very protective bacteria in their gut.
Yeah.
Their gut bacteria have like evolved a way to resist it.
Hmm.
So this paper kind of focuses in and looks at kind of what isolates can resist like attacks by the cholera's type 6 secretion system.
Yeah, like in
And
figure one, they just have all these numbers of random bacteria that they've isolated. They only give them numbers, right? But they're comparing their ability, their competition, right, essentially with cholera.
yeah, yeah, they do like some competition assays. So sticking cholera next to another bacteria and see which of the bacteria survives.
Yeah.
And they found like that there were like some that were really kind of good at surviving.
Hmm.
So
And
let me see
and
whether
there are these
yeah.
theories. So again, we haven't really dove deep into a type 6 secretion paper, but there's this idea that like type 6 secretion, there's antitoxin systems, meaning that the toxin that the bacteria wants to be is deadly to other bacteria, but it's not deadly cholera because they have an antitoxin. And so
Hmm.
they use the type 6 to inject the toxins in, but there could be an arms race that emerges, right, where there's value in keeping random antitoxin things around inside of the bacteria that don't want to die from getting injected by a type 6. But this paper tries to focus on other factors that might influence that. They don't want to look into that whole. It's quite a complicated world of like basically different overlapping relatednesses of toxin antitoxin systems that would give susceptibility. They're sort of looking outside of that to find some other factors. And I think they do
Yeah.
end up finding those other factors. What's a good figure for that?
Yeah. So, I mean, they found all sorts of stuff. They found like one strain that actually kills off Vibrio cholerae using its own type 6 secretion system.
Yeah,
They also find that some of them secrete like I think almost a
a
capsule or
capsule.
something like that.
Yeah,
Yeah,
figure.
that
Oh gosh, I can't even see the figure number on this one. Oh, figure four. Figure four, they find some stuff that, yeah, they seem like they're being protected, and they 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 attribute that to the type of capsule it has. They even do like a genetic mutant that doesn't have like some of the capsule biosynthesis, the WZA, and they find, yeah,
yeah.
like this is the protective factor. So there's many. I guess the thrust of this is that there's many different ways, right? That all these different bacteria protect themselves against getting injected by type 6.
Yeah. So like, I feel like capsule is the equivalent of like of holding someone away from you with
Oh
your
yeah,
hand, and
yeah,
then they
yeah.
can't punch you. That's kind of like
I like how you made it so physical, but
like
like I was just thinking a force field.
yeah. I was just thinking on the playground when someone shorter than you tries to attack you, and you just like put your head under the head, and they can't reach you.
Yeah,
That kind
yeah.
of
No,
thing.
that's great. And again, like this is I think I love this about microbiology. It's like we're working at these scales that are really small. So like making them macro like actually does give us like this good sense. Because like what you described there is like a perfect physical analogy, right? Like capsule is like a large molecule, a large polysaccharide molecule that's like blebbing out from the right the surface of these cells. And type 6 is this protein spike, essentially a needle
Yeah,
that needs to get close. And so if that needle can't get close, then you're not getting injected.
yeah.
All right, that's
Um,
all I think I want to say here. So like again, like you see a lot of type 6 research like related to this sort of ecological, right, like niches that bacteria need to live in. If people are interested in that, let us know, because like I don't think we have done a good deep dive into these sort of systems and how they impact the types of assays that are used and things, these competition, so many competition assays.
yeah. And speaking of bacteria fighting other bacteria, I'm quite interested in this next paper, which is engineering a genome-reduced bacterium to eliminate Staphylococcus aureus biofilms in vivo.
Yeah.
So
So I just want to
yeah,
take
this
a
is
nod to, I think we saw like a wacky technology to try to eliminate SARS-CoV-2 where they made like defective SARS-CoV-2 viruses, right? That like they
right,
take up space and they're unable to produce productive infection. And we also saw that in some of the weird tripartite interaction with phages, right? That like that's also a way in which they try to take up space. Like if they have some sort of genetic element that gets in there and like makes a defective virus. But this is the same thing almost in bacteria.
yeah, it's interesting because it's both same and kind of because so firstly, what this mainly describes is the creation of this genome-reduced bacterium, which is based off of Mycoplasma pneumoniae.
Hmm.
And the idea behind it is like that using a bacteria and altering it to give it all the tools to to kill off Staphylococcus aureus by producing various dispersants, biofilm, and also to like have it take up resources, I guess.
Yep.
Um, yeah, Staphylococcus aureus biofilms are real problems. They can form on like was it artificial like organ implants or
Yes.
vents. That's a big problem for that.
I mean, just as you
And
were talking about like diseases get by like the types of things they do. Staph aureus has like a whole suite of things. It does lots of things. Most bacteria, I think, do, right? They have much larger genomes. They have much larger virulence factor like arsenals to be able to get in different niches.
Oh yeah, I mean Staphylococcus gets associated with respiratory infections. It also causes impetigo as well. It causes a
Hmm.
form of toxic shock, wound infections, pimples.
Yeah.
It's got a big CV of infections,
Yeah.
and yeah, I think it is interesting because when you come from a certain area of science, you do tend to like get limited to that. I remember because I worked on strep, so I got very limited to the idea of like one certain like model of strep, but actually does so much more.
Hmm.
So,
Hmm.
but yeah, anyway, the interesting thing here is the mycoplasma. So I don't know if you heard about a few years ago when like the Venter Institute like created their own minimally reduced like
Yeah,
genome,
that's
right?
right. Hmm.
Yeah, so that was based off of mycoplasma because mycoplasma are very like small bacteria that have very small genomes, which means that people have been trying to create like a minimally functional genome to like essentially their own bespoke bacteria that can do what they want. And
Yes, yes. This is definitely an engineering thought coming out of synthetic biology, right? They want a chassis. People want the perfect chassis. Like a
yeah,
nice frame, Model T frame that they can add all doodads onto.
yeah. The one problem of using Mycoplasma pneumoniae as a treatment is that it causes a disease in humans, which is a pretty bad thing to have if you want to use it as a treatment.
Uh,
So quite a lot of this paper is dedicated to detoxifying it, taking out the things that make it dangerous to humans,
yeah,
and essentially minimizing that genome, and
yeah.
then adding in a was it a gene called dispersin B, which is able to break down the biomolecules, the kind of very the basic building blocks of biofilms.
Hmm.
Um, and yeah, so that and another big part of this is they use PET scanning to look at how the biofilms get destroyed, which is
Hmm.
a really interesting technique.
Yeah, I don't think I've seen PET scanning to look at biofilms. Seems like such a complicated piece of machinery, but I
It
guess
is.
yeah.
It, I mean, the good thing about PET scans is you get pretty good signals out of it. The problem is you're not necessarily sure where those signals are coming from,
Hmm.
because in this case they use FDG, which is the metabolite that is taken up by biofilms and also immune cells and other kind of host cells as
Mm.
well. So when you're
Hmm.
analyzing like data using PET, you have to for something like this, you have to have comparisons. You have to have your very tight controls. So
Right.
uh, in here, what they do is they actually insert like a tube with the biofilms in them, and you can see
Hmm.
it kind of like apparent under the
Yeah.
mouse's kind of skin, and
It,
then
it's a model for like catheter infections, right?
yeah,
Like, and so they're saying, oh, here's some plastic that has a biofilm on it. Like, can we show that, like, if we, I guess, pre-colonize them with something else, that that biofilm will get disrupted? Or sorry, not it's not even pre-colonized. It's like this is like another invading bacteria into the pathogenic biofilm.
yeah, and that is what they do. So I'll scroll down to like so. There's one figure where they show what does to these tubes outside of the body. So they've got
Hmm.
like a tube with like the like so they've got a tube where they've just got the staphylococcus forming biofilms, and one where they've added their mycoplasma that produces dispersin B. And you can see that there
Yeah.
is a definite reduction in biofilm there. Um,
Right.
and they try to monitor it in vivo because obviously they can't do that same thing. They do like non-invasive imaging. So we saw a bit of that not last week with bioluminescence. Here they use
Hmm.
PET. So PET essentially you have a radioactive marker that produces a positron that gets annihilated and sets off some light. Um,
Yes.
and they got these like detectors like that capture those positrons and figures out the direction of where they're coming from. And based on that, you can try and figure out where the signal where the signal's origin is.
Yeah.
Um,
Yeah, this is
yeah,
a wild technology. And also like it's all done in mice here. Like you imagine that if anyone wanted to translate this technology into humans, like you'd have to redo everything, right? Like
I
you got
mean,
to make sure the chassis doesn't hurt humans, like because they're injecting this mycoplasma just into the right
yeah,
into the mice. Yeah.
yeah, they need to. So there are a couple of like security things why they prefer mycoplasmas because partly because its genome is set up quite differently from other bacterial genomes. So the
Hmm.
idea of like some genes being transmitted between mycoplasma and staph is quite different because essentially the genome code reads out differently So it reads out
Yeah.
different proteins and different stuff. So things that would be like normal for mycoplasma might end up at stop codon. So there's that kind of security there.
Hmm.
It's a lot smaller, but yeah, this is very much a toy example. Um,
Yeah.
it's not anywhere near ready for like clinical application, but it's kind of
Right.
almost a proof of concept
Yeah, it's like
that
it's an interesting idea, and like talking about like all the different I love thinking about all the different ways biotech could be used, right? And like
right.
this is like yeah, this is this is definitely like on the very far distant future side of it, right? Like bespoke bacteria that do the thing that you want it to do.
Yeah, yeah. And yeah, I think that's kind of what attracted me. That the because yeah, it felt very futuristic with their radiation and bacteria they program
Yeah,
themselves.
pet scanning.
Yeah, pet scanning to scan your pet mice. It writes itself.
Okay, cool.
Cool. Next paper is I like this one. Bacterial quorum-sensing regulation induces morphological change in key host tissue during Euprymna scolopes Vibrio fischeri symbiosis.
Yeah, we haven't talked about yeah, Euprymna scolopes or the Hawaiian bobtail squid, I believe is a
Yeah.
cuter and name. It is a very cute animal, like a very
Yeah, yeah.
tiny. Oh, here we have some baby images. Yeah, this has been a model for like bacterial symbiosis for quite a while. They have a specific organ in their bodies that when they're born, it doesn't have anything in it, but from
Mm.
seawater, it concentrates a specific chemoluminescent bacteria that gives it a glowing, that allows it to glow. And people think that it gives like a light. So like during moon, they feed during the night. So like the idea is that their luminescence camouflages them from anything that wants to eat them from below, or something like that. At least that's one of the
Yeah.
functions that people attribute to this organ.
I mean, yeah, because bioluminescence is actually really common in deep sea. I think
Hmm.
there's a lot of different like organisms that either are bioluminescent or even use bioluminescence.
Yeah.
So, I mean, I think like one of the theories of why killer whales have those white spots across their head is because they're hunting bioluminescent organisms. So when they like kind of it helps them see like the same way that like people put like black like eyeliner when they're like playing football so they get blinded by the sun. It's like kind of
Yep.
the reverse.
Yeah, the reverse of the black eyeliner. They do
Yeah.
want to blind people in this area. So,
Oh, even like great whites where they're like they got a white underbelly and a
yes,
black top. So
that's actually a really good one. Yeah. So yeah, this is what the squid does to do it, and it's really awesome for studying symbiosis because here's like an organism that's co-evolved, right, to have this super close relationship, right, with the bacteria that live on it. And so in this particular paper, they are looking at something that when bacteria grow together, right, they want to sense that they've hit like a critical point, and
mm,
that's called quorum sensing. They make a molecule, it's concentration dependent. The more of it around, like basically induces like a positive feedback loop. But what's fascinating, right, when we're studying symbiosis is those bacteria to bacteria signaling can trigger, right, the expression of something that then affects the host. And that's what they're investigating in this particular paper. Um,
yeah.
so
So
yeah, let's just I want to see like yeah, if we scroll down a little bit just to get a sense of like this organ, right? Like
yeah.
yeah.
So
Here's
they
like oh yeah, go ahead.
yeah, no, they've got this like organ, I think it's called well they've got light organ and inside they've got like a little like they got a little channel where the bacteria migrate through. And the
Mm-hmm.
idea is like when the bacteria like go in, they get loads of nutrients and they decide to just settle down and start a family there. So
Yeah.
they produce quorum sensing, and so they produce these molecules that let them know that there's lots of bacteria here. Let's all just start lighting up.
Mm-hmm.
And another thing they also do is that according to this paper, they might send signals to the host to let the host know, here's you've got lots of bacteria now. You don't need to take in any more.
Yeah. Yeah. And they look at that from the developmental standpoint of the squid and they relate that back to this quorum sensing. So yeah, I think if we go to the next figure,
Yeah.
um
See. Yeah, so they've got these channels that let the bacteria in, right? They're
Mm-hmm.
called so I think like let me see whether
Oh, I
um
don't remember what they're called.
because I remember like they call like the bits where the bacteria colonize crypts, um
Mhm.
and I
Yes.
think like yeah, so
And
each side of this like will contains like crypts.
mhm. And in the when they make these mutations, when they make that Lux L mutation, that's one of the quorum sensing signals. They can see that the bottleneck, the pathway into the crypt is getting narrower.
Right, yeah.
Or wait, getting wider because there's something that's being made in the wild type form that narrows the bottleneck. And this is what you're saying, like, because once you have something that's established, the idea is that the squid doesn't need anything more, right? Doesn't need to bring any more bacteria into that environment.
Yeah, I mean, if it keeps that bottleneck open, it opens it up to like, say, other bacteria coming in, or even a phage destroying all their precious bacteria. So
Right.
having this, so they shut off their bottleneck and this keep these bacteria with them for the rest of like their lives, I presume. Um,
Yep, exactly. Yeah, so like just like a really fascinating story from that perspective. And I love these, I'm a big fan of these things because it shows us how closely linked we are, right, to the fates of bacteria. And I think this is also what drives people's fascination with like our gut microbiota, right? And
yeah,
everyone's crazy about gut micro like brain axis or whatever because you look at nature and you see these examples, right, of some pretty major changes to the physiology of the animal from the bacteria that goes in. Of course, in this case, like these bacteria are like so critical for the survival of this animal, right? This organ is dependent on getting the right bacteria in and keeping them there.
yeah. I mean, with humans, you've got like very much a maternal like transmission of bacteria as well as lifetime of like gut
Hmm.
microbiome. But I know that there have been experiments with like mice where if you don't have your gut microbiome, bad things happen and things that you wouldn't expect would
Oh
happen
yeah.
as well. Um, I don't know those off the top of my head, unfortunately, but just, you don't have to take my word for it. Just like, yeah, but
Yeah, it's immune system stuff,
yeah,
right?
immune
Like this is
system
like
stuff.
how no notobiotic mice are like really like yeah, they don't have a very nice immune system.
Yeah,
But
um,
yeah, I think that's all I really want to point out in this. Like the rest is just the proof of like right after seeing that picture, right, of a narrow bottleneck versus a not so narrow, then they just go in and they try to show that through various mutants that they
well,
have.
yeah, we got the nice picture. Now they have to like pick out exactly what's happening. So what part of the system is
Yeah.
it? Like, because you might think, oh, maybe it's just the light. If they get bacteria that aren't producing light, maybe they've got some light sensors that say, oh, now we can close the bottleneck off. Or like, is it a
Yeah.
specific like part, other thing that because these bacteria have lots of core processing systems, so they want to make sure they've got the right one. So
Hmm.
yeah, there's a lot of
Hmm.
other extra work that goes into this.
Yeah. Okay.
Ah, yeah. Next, we're having a bit of MythBusters on our show, like the the myth of antibiotic spider silk, where these group of researchers are doing a lot of debunking on about the
Yeah.
scientific literature. Um, so I think like let me see because I got some choice sentences here, like where they say like this notion that the antibiotic spider silk like because this idea that spider silk could be an antibiotic. I mean, it makes sort of sense, I guess, because I
Yeah, I
don't know
guess
why, but
like
yeah,
no, maybe because like
yeah,
spiders want to keep a clean home or something like
yeah.
that.
And spider silk is bulletproof, and so therefore, what else can it do? It might be magical. But yeah, apparently, like this notion is based on studies reporting antimicrobial activity in spinal silk. But, and this is I'm quoting from the study, close inspection of these studies reveals that the evidence is conflicting and at best anecdotal, and that's a burn. Uh,
Totally. Nothing worse than having your scientific paper called anecdotal.
yeah, and so they've got a nice picture of a spider. Oh, yeah,
Busted.
just. I mean, this is they're living the dream of a lot of scientists. I think just
Uh,
busting
Duncan
some.
on Duncan on other scientists using data.
I
I
mean,
mean
look,
that essentially is what happens in all
yeah,
papers.
I've got like literally like a paragraph like so I've got this table and I really like this table because I got this table very similar in my PhD thesis of just showing all these references going back several years of all the other people who study this and then listing what went wrong in that study and why they can't be trusted. So
But
like,
this is, I mean, these are great papers. It's like it's interesting, right? Like, what brings like some of these papers to the forefront? Like you said, you did this in your thesis, right? But people don't get really, not many people review those.
oh,
Um,
I did it because I got a bad review on one of my papers, and I thought, and I was so upset about the review. I was like, okay, here's my response in my thesis because of like 50 papers proving you, mysterious review I don't know, wrong, and so it was very much a psychological thing for me rather than
Yeah, but no,
it's not
but there's
yeah,
value to that type of there is value in that scholarship, right? Like having that in
yeah.
your thesis, like if someone did want to dust off, right, that in the archives, and then they instantly are situated in the literature in a really meaningful way that relates to the results that you're presenting. Um,
Yeah,
it's just I think that like again, like there's some balance sometimes, right? Or first of all, like is that hot science, right? Like just like doing these types of reviews and uncovering things. Sometimes that's not as maybe compelling as others.
I mean, I think it's a really important because the idea is that peer review should filter out these studies, but sometimes peer
Yeah.
review doesn't necessarily because peer review is just two people reading a paper like us,
Yeah.
and we make errors all the time. So,
Hmm.
I mean,
Hmm.
yeah. What does that say about the rest of academic literature? So there's some stuff that's gonna like flow through, and I think there is a
Yeah.
lot of value for people who will look at the literature like with a critical eye and point out
Totally.
things.
Yeah,
Um,
so I'm glad that in this paper, I guess the excitement of antibiotic spider silk warranted a little bit of a takedown in table form.
Yeah. So yeah, I mean, I think this table is a nice like kind of primer on how to like synthesize information from papers because I think
Hmm.
especially with the way we go through papers is we go quite a lot like through quite a lot and we have to skim over and don't get a lot of the details. And this one they actually like pin down. Okay, here are the issues. So for
Yeah.
instance, like risk of bacterial contamination is a big one. So this, I mean, the first like I'm going to highlight this because essentially what they say is like, oh, they didn't mention anything in the paper, but in the thesis for the work reports that the samples were highly contaminated,
Nice.
and but it was assumed by the author that it didn't influence the growth of the test organisms, which they don't know because they didn't have like a control
Hmm.
to test out whether because anything can be a contaminant, like penicillium could be a contaminant, and if that's a
Right.
contaminant, so it's um and
Yeah, no,
another
that's a great example, right? Just to highlight that like the devil is in the details here. And that's why like yeah, it's great to, chat about papers and try to get high level like what's going on, but until you go into the methods, like you don't really know.
Yeah, I mean, we try, but I think it takes a lot of expertise to. I mean,
Yeah.
oh god, my favorite one is unclear how the silk was obtained because I think a lot of these people went out into the like environment and just picked up some spider webs and assumed they were of a certain species, um,
Uh-huh.
which I guess could make sense if you're, but it means that like it's harder to replicate what you did.
Yes.
Um,
Yes.
yeah, and another thing is like the diluent. So sometimes when they extract spider silk, they have to dissolve it in something, and that's something in this case I think was blah, blah, blah. Well, what they did dissolve it in was oh yeah, there is ethyl acetate, which is also dangerous to bacteria. Um,
Hmm.
so it's so in this case, these sort of cases, you have to compare them because if your diluent can also kill off bacteria, you have to make sure that any antibiotic effect isn't due to the diluent. And I've seen people have mental breakdowns that were trying to get controls of the diluent. So it's
Sure.
um um oh wait, hold on a minute, and I think they did a spelling mistake. So yep, abs Rob, ha. So guess nobody's perfect. Yeah, suck it. We're better.
Yeah. So they essentially say like given this like sort of uh intense methodological error that's being propagated through the literature, like let's try it again.
Yeah, exactly. So yeah,
Let's
they
try it. Yeah.
and so now they have to do it really well because otherwise it would look really embarrassing if they made it, just from this. They have to make sure they don't make the same mistakes that
Right.
they pointed out. So I think that's kind of a great little like tightrope walk um
Hmm. And it's
yeah,
pretty simple actually when you think about what the experimental design is here. It's just that probably doing it well does take like a very careful, right, a very careful hand, and yeah, and making sure including the right controls. So they collect silk from many different species, from many different types, um,
Yep.
and they get something to dissolve it in, and they do a classic antimicrobial um, resistance assay.
Yeah.
Little
And apparently they don't
discs with
they
it.
they work with all these different species of spiders and they don't seem to suffer from any heebie-jeebies at all, which is
These guys love spiders, man.
yeah, they um so yeah, and so this is I think this is an interesting paper just to read so you can see kind of this analysis of literature and the way people analyze it and add to it. And also just for the sick burns, I think is quite a fun paper. Um,
Yeah, don't say there isn't any drama in science. It just sort of takes place like silently in papers and over many years.
yeah, they it's all very like kind of prim and proper. We don't actually go, "Oi, f you," yeah. But yeah,
Okay,
ah,
so moving on, we got this paper in eLife, Adaptation and Compensation in a Bacterial Gene Regulatory Network Evolving Under Antibiotic Selection. So we talked about antibiotic selection, right? Like you have this selective events. It means that random mutations can accumulate, right? The things that survive those and maybe are a little bit better, like they go on and they might get evolved again. But this looks into, I guess, sort of more of the nitty gritty of exactly what those mutations are doing and what is the nature of the mutations that occur afterwards. So they look at a particular model. Uh, the antibiotic is called, I've actually,
trimethoprim.
yeah, trimethoprim, which I hadn't heard of before. I'm not like a antibiotic expert there, but it targets a specific set of like a two-component regulatory system, and then also like one of the I guess like metabolic genes that like then govern
Hmm.
the specific resistance. And they find that the mutations that end up occurring, the ones that occur later are sort of modifying the that initial resistance mechanism to make it more, I guess, to be more harmonious with growth in the
Yeah,
bacteria. Yeah.
because I think like the initial mutations are just so the bacteria can survive, but its growth is slowed down, and
Hmm.
there needs to be all these compensatory changes. So it's not because each gene affects is part of a much bigger system. I mean, two component systems are the main communicating systems in bacteria.
Hmm.
So um, so like in if you mess up one, then there needs to be a compensate, and so they look at basically effectively like I think the evolutionary choice of transitioning from like being like tolerant to being tolerant of it, and then going down this other way. So changing, it's almost like redoing the way it does its metabolism in response to trimethoprim.
Hmm.
Um,
Hmm. Yeah, I think what was something that I thought was interesting. I don't think it's in figure one. Um, they find like a concentration dependence, which is kind of interesting. Oh
hmm,
yeah, uh in figure three,
right.
in figure three, they're like looking at these different concentrations um of the antibiotic, and they're seeing like, okay, in this when they knock out the certain genes, like how does that affect the bacteria? And there's like there's a break point, I guess is like what to take away from this, where that if you have I'm not sure. Maybe I'm not actually well equipped to explain this one, but I read this in the abstract, so this is me like not diving into the figures enough. um they do talk about the fact that if you give more of the antibiotic, you'll induce different types of mutations.
Right. Yeah.
And I guess like so I guess it speaks to what I wanted to have people take away from this. It does speak to like people say like you should take your full course of antibiotics, like so that you're just eliminating everything. Like if you have low amounts of antibiotics, you're giving bacteria the space to start doing other changes. right? That also might help. So it might not just be that like the initial chance of getting a resistance, like it's not just like the initial emergence of resistance is important. It's that like even the ongoing the life of that resistant bacteria is like honing its ability to be, you know, a successful antibiotic resistant bacteria. And so taking like that high dose, I guess, of antibiotics through a long amount of time is really eliminating any chance of that.
Right. Yeah, I think that that's interesting. I think we can move on to the next paper because I don't think I've made much to add to that. Which is a paper about uh, Basalt-Hosted microbial communities in the subsurface of the young volcanic island of Surtsey, Iceland. Um,
Yeah, I think
yeah,
we saw like they did like we've seen a back thing where they did ice cores and they had all this like complicated thing to like see the bacteria that grew in ice cores, make sure they sterilize the outside and all this. And now they're going to do it in a rock.
yeah, yeah. Because this is like a. So I don't know if people are aware of the story. See, Surtsey is a new island that appeared in the '60s out of nowhere. Like there's a volcanic eruption. It produced this island, and it's been interesting for science to see like how do new species colonize it. Because it is just completely new land that,
Yeah.
and it might be interesting to learn about how those processes work. But another thing that people don't really that we don't really focus on when we're talking about Surtsey or anything is just uh, microbes in the. There are lots of microbes in like the Earth's crust or like
Hmm.
a subsurface microbiome that we don't know very much about, and so we know there are like bacteria that live like in rocks underground, and they might be 15% of the total biomass on Earth. And we don't get any opportunity to investigate this biosphere, especially the deep biosphere held like hosted underneath the ocean crust.
Mm.
But Surtsey comes along and just vomits up a bunch of it. Uh,
Yeah.
so it's an opportunity to investigate this in this like kind of niche where that bacteria can survive in.
Yeah, absolutely. If we go to figure one, you can see where it is in the world. And yeah, they also show you like the boreholes that they ended up drilling into it, like where it is on that new chunk of rock.
Yeah. Okay. Yeah. So there's see the south coast of Ireland, and then you've got Surta, like Icelandic, like fire god, I think. All I remember is because I just watched Thor Ragnarok recently, and I'm like, oh, I know what that person is.
Yeah, so they drill down these cores and they do 16, you know, the classic 16S sequencing. So they have to extract DNA from rocks. Um, and they get a bunch of hits. And of course, they have to do a complicated in figure two, they have to do a complicated like cross comparison of all the different potential contaminants that they have in order to narrow down like the things that are only in the drill core because of course like
Right.
like they it's difficult to make sure that those drill cores are free of any of these other things and they want to know like what's living in the rocks. That's their interest here in this paper.
Yeah. So essentially, they're looking at so the bed in orange, and then there's all this. Yeah, it's quite a difficult experiment to do.
Hmm.
Um,
And
because
and
yeah,
they find a bunch of things, which is interesting. And I guess like I don't really have anything specific to say to like what things they find. But the idea is that this is a description, right, of of a
mm,
bacterial environment, and maybe we can compare it to other things, or we can say something about the metabolism, right, or the types of things that like to live
yeah.
in rock.
So this is kind of like data that hopefully will people can come back to and say, oh, we've spotted this enzyme here, or we've understand how the system works. So this is almost like building the boundaries for future research.
Hmm.
Um,
And I think
because
the
yeah,
figure that I really enjoyed looking at was figure six. And like because I mean they found bacteria that live there, and so they also did EM of some of the rocks that they removed. And you can't tell, right? They don't correlate this to anything. Like I
mm,
think, you know, we've seen this before. Like you can take EMs of things, you don't know what it is exactly, but they want to just speculate a little bit about things that sort of look bacteria-like, and as a way
yeah.
of maybe bolstering some of their claims for bacteria being in these areas.
Yeah, I mean it always looking good at EM, but I am reminded that Mars meteorite where they did EM on something and it looked like a bacteria, but nobody
Right.
knows.
Exactly.
But,
Well, I mean, they just have to do the DNA extraction. But I guess how do they? There's all these weird, right? Like, if there is extraterrestrial life, like, how do you know that it's going to come up in DNA, right? Like that's sort of what's said. Yeah.
exactly.
So here we can like cross
Like,
confirm because this is Earth. Like we sort of know what's going
yeah,
on Earth. But yeah, it's not.
but yeah,
It's
I
only a
mean,
cool picture actually, and not any definitive evidence.
yeah, I mean, it'll be cool if in future someone goes, "Oh, wait a minute, I recognize the shape of a bacteria that we found in rock," and then they go back here, and, oh, it's very similar.
Yeah.
But sometimes, yeah,
I mean,
it's an interpret.
I'll already
Yeah,
say that like
I
there are some things here that like they're pointing to that they say like, oh, this is like what clay looks like. So like, how do you right like you have to have like this very you have to have a really good knowledge of like all the different ways things look like under EM in order to say like, like these things look like bacteria. Like I will say like those things are EPS like, but like I saw it also in like in other cellular material and not even alive cellular material, right? I've seen these patterns in dead cellular material. So like there's
yeah,
another sort of question to be asked there. Whether
yeah,
there's
for sure.
viable bacteria in these areas or like right like but maybe there are viable bacteria but they live in a specific area, then they get like these could be kind of like fossilized in some ways, right? Like then they get like spewed out right upwards into the right into this new island, into the rock, and they're just dead. Right, they're dead and their DNA is still around for some reason, right? But like they aren't doing anything.
Yeah, I mean, it could be some form of bubbles created by the volcanic reaction that produced Surtsey, but
Yeah.
I mean, it's difficult to because again, I think this kind of subsurface microbiome is something that I personally don't know very much about.
Hmm.
So it could be that these look similar to something, and I feel like there isn't that like level of background knowledge.
Yeah.
So if we find lots of DNA genes, there might be a lot of unknown stuff there
Hmm. Hmm.
that we don't know what it's
Hmm.
for. And it could be that we might be looking at things that have very different metabolisms than what we're used to dealing with,
Yeah.
or different way. Yeah.
I just think
So
it'd be cool,
it's
like in
interesting in
the.
that.
I mean, I don't know if anyone will ever do this, but if people do like a funny model, right, where they try to grow some of these unknown bacteria, right, like grind up some of these things and put them in an environment they think it would grow, then you'd take pictures and you'd be like, oh, those are like the things that we saw when we took pictures of the actual core samples. But I mean,
Yeah.
that's going to be
And
so far away because I can imagine like there's significant challenges to culturing something that wants to grow right in the Earth's crust like this. So in the Earth's
yeah.
crust.
I, I think I read somewhere in there that there were lots of thermophile like kind of genes as
Yes.
well, because I guess at the time when, because I've assumed that volcanic probably the microbes has to be used to things that are hot. So and it is an active volcano still, if I recall
Mm-hmm.
correctly.
Mm-hmm.
So um, so yeah, there's a lot, and of course, like thermophiles are really interesting for
Yeah.
bioengineering
So they didn't
and
do
for
so in this like it's like again, this is like such a cursory survey. They don't do like any like sequencing of the genomes themselves, right? This is just like this is
hmm,
just like a list of all the things that grow. And they find that that list overlaps with
right,
things that are thermophiles.
yeah.
Yeah.
So it's interesting. It's very much a first step,
Yes.
but I think
Yes.
that I hope you lathered this paper. Uh,
Yeah, and that's all we have for you today. So join us next week where we will dive into like a more cool microbiology news.
Yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. We think that peer review is a process that we can all participate in. So we hope that you've had a good time listening to us ramble on about microbiology today. If you have something to add or found something unclear, just let us know in the comments.
Yeah, it's been a pleasure chatting with you,
Same here,
Danny.
Paz.
Tune in next week for more microbiology content. Bye.